Or live and direct at ICCS 2024, Seattle, Washington. Gage Black, bioinformatics scientist, is in the building. All right. All right, it's break it down for
All
us.
right. So today we're talking about our work, batch normalization, specifically in mass cytometry, but also plus applies to spectral cytometry. The problem is every time you run your samples in different batches, you're bound to get some type of technical variation applied to your study. This is looking at the expression of the different markers within the cell. Cells are clustered together based on these expressions. But unfortunately, in reality, you don't actually get this perfect overlap. You have some type of technical variant introduced. So that's what you're here, where in this population right here, you see more of the red cells over here, which represent batch 3 and 4 compared to these down here. Really, that as you have more and more batches, the more likely you are to introduce variants. Where need for batch normalization comes in, and where we are comparing two common batch normalization tools, one called Cytonorm and one called Cytomind. Both are packages that are available. And through this you 38 patients at Keystone General Hospital, and we analyze these using three different. One is the reduction of urban thickness, and this is a calculation of the reduction in the technical variant. So the closer to one you get, the more perfect you score it. The next is the median absolute deviation, or the MAD score, which is a measure of the biological variance that you were able to retain. So the closer it is to zero, the less biological variance is put out. And then finally, the last metric we were comparing is the percent efficient variance right within each cell type across the different batches, seeing how much variance there is. So when we look at these results compared to non-light, both tools do a very good job at maintaining 0.03, again, really close to zero, meaning the biological variance really is being maintained, which is great for your study. And then we're next looking at the EFV, which again is that measure of the technical variance that you're able to reduce in the sample. That Cytonorm has a reduction of 0.37, and like Combine has a reduction of 0.7. Again, closer to one means perfect. Combine actually does a superior job at reducing the technical variance data. Because of this, we were excited about Cycombine, but then we took one step further, looking at the percent CV. So again, looking at each cell population and seeing how much variance there is across the 4 batches. That's specifically done in our control samples that were run. We actually found that Cycombine does not have significant reduction in the percent CV. And when looking at the cell populations as a whole, we actually don't see any significant reduction. We do see that the percent CV is lowest when you're looking at the more common cell populations. As cell populations get more rare, the CVs start to go up. But we took this one step further, and we decided to look at functional markers and seeing how the expression of these functional markers for froth batches. We actually see that Cytonorm does have a statistically significant reduction in the percent CV on average when looking at these functional markers. Because of this, it's exciting that Cytonorm has this reduction, but Cycombine seem to be having greater technical variance reductions. We conclude that really it's a complex issue where you need to really understand your data. You need to look further. You can't just rely on one metric to determine which tool is best for you. While batch normalization is important when you have multiple batches on your study, it's important to look at these different metrics and really determine what's going to be best. And that's something we're going to continue to dive into and figure out which tool is best for each data set.
So, Gage, would you say this is generalizable, or is this data set specific? How
Yeah, that's a great question. So the need for batch normalization is generalizable. It's very rare that you're going to have a perfect run across multiple batches. I think that the results here are applicable to this data set, and it's something that we want to look further to see how the numbers change as you look at these different data sets, because each sample is going to be different in its own way. Each indication is going to be different. It's really hard to give one very clear answer.
Absolutely.
All right. Thanks for joining us today here at ICCS. Great time. Go flow cytometry.
Awesome.